The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining me today in this webinar on healthcare processing and how to design an IFU and validations for Europe and the US. My name is Katleen. I am a study director for Nelson Labs, and so in my function, I help our customers with the design of their cleaning, disinfection, and steam sterilization validations. And what I found in my work is that healthcare processing can really be a challenging field to navigate. There's a lot of different hospital practices that are being used in different regions of the world. And also, there's not a ton of guidance out there on how to design your validations, but the guidance that we do have also is different in different regions of the world. So this can really be a challenge when you're designing your IFU or when you're designing your validations. So instead of one straight path forward or one right way that you can design your validations, there are several options and all of them might be right. I tend to compare this a little bit with going on a road trip where you can make different paths for yourself. It just depends on what attractions you want to see along the way. And so it's really the same for healthcare reprocessing. You can do different things that might all be right. It just depends on what your market is. So what I want to do with you today is help you navigate this landscape of healthcare processing and help you design your road trip in the IFU and in designing your cleaning validations or your disinfection and your steam sterilization validations. So I want to be your navigator in this process. But before I start, I always like to just very quickly take a step back and go over the basics. So I want to talk about what actually is healthcare processing. Why do we need to validate these processing instructions? And what kind of devices is this applicable to? So when we look at this picture of a surgery being performed, you have, of course, all of these devices and materials that are going to be thrown away after use, but there's also going to be a lot of reusables in there, such as surgical scissors, or you see these overhead linings as well. They are going to be reused for a next patient. And we want to make sure, of course, that they are safe when they are being used on the next patient and that any contaminants that were on there have been adequately removed. And that's where healthcare processing comes in. So here you can see the major steps in the process. You have first always a very thorough cleaning step. And then in the second step, we're going to do disinfection and or sterilization. So important to understand is that these are really distinct processes that have different end goals. In cleaning, what we want to do is we want to make sure that any soil residuals that are present on the device have been removed. So we want to remove blood, we want to remove proteins, we want to remove tissue, we want to make sure that all of that organic debris has been removed. And then in our second step, in our disinfection and our sterilization, that is where we are going to kill off our microorganisms. Now, historically, a little bit more focus has been put on this disinfection and sterilization because, of course, microorganisms are what's going to actually cause healthcare-acquired infections. But we now know that if your cleaning is not effective, your disinfection or your sterilization is also not going to be effective. So these are really very important processes to have. Now, as a device manufacturer, it is your responsibility to tell people how they should process your device. So you have to design the IFU, outline the cleaning steps that have to be performed, or the disinfection or the sterilization steps, and really outline those instructions in your IFU. But of course, it is going to be the healthcare personnel that's actually going to do these instructions. And they are actually going to perform these processing steps. And on a day-to-day basis, what they will mainly have access to is a visual inspection. So they are going to perform cleaning, look at the device, and see if it looks clean to them. But that, of course, doesn't mean that it is clean or that it is clean enough. And that is why we need to validate that on beforehand. So that is where, for example, Nelson Labs, as a testing lab, we are going to not only look at the device, but also quantitatively assess how much protein is still on there, how much hemoglobin is still on there, or quantitatively look how many microorganisms are still on the device. So that is why we test this on beforehand. Now, there are three main categories of devices that need these processing instructions and therefore also these validations. The biggest category is the products that we mostly think of when we think of reusable medical devices. So these are your surgical tools or endoscopes or dentistry tools. So any kind of reusable medical device. The second category is actually single-use devices, but that are processed by the hospital once before use. So the hospital is going to clean and sterilize them, for example, once before they use the device. The third category of the devices is home care devices. So these are often single user, but they are going to be processed at home for another use. Typical devices that you can think of here are, for example, insulin pumps or breast pumps or menstrual cups. So the things that people really disinfect at home. So with that, let's start our journey. And let's start by looking at cleaning and the sort of different crossroads, let's say, where we might need to make a different distinction between your markets. First step is the cleaning IFU, and there are two things I want to talk about here. First is the cleaning method, and the second is the detergents. So here, both of those steps, whether your target market is going to be Europe or the US, you may need to take a different exit. In terms of the cleaning methods, so you have two main cleaning methods. The first is automated cleaning, where we use a washer disinfector that's going to do the process for us. Or we have manual cleaning. This includes steps like immersion or brushing or flushing. It can really be a variety of instructions. Now in Europe, if your device can withstand automated cleaning, this is something that you have to do. So it's not optional. If your device can withstand it, you have to have automated instructions for cleaning in your IFU. In the US, there's not a very strict requirement. So it's not that you have to do manual cleaning, but we do see that manual cleaning is often still preferred in the US. Now, of course, what does that mean if you want to target both markets? So if you want to target both Europe and US? So the first option you always have is to do both, right? To have both a manual cleaning and an automated cleaning procedure in your IFU. Now, of course, if you want to have both in your IFU, that also means that you have to validate both. So that means more time and more money. So because for Europe, automated cleaning is really a must and it's really non-negotiable, our advice is that if you want to do both and you only want to do one, go for automated cleaning. If you have more budget, you could think about doing both. Because also there might be hospitals that don't have access to automated cleaning. And so you might really expand your user base by doing a second cleaning step. Second path where we might take a different decision is the detergent. So you have different kind of modes of activities for detergents. You could have detergents that are mainly neutral pH enzymatic detergents. And then you have a big class of detergents that's alkaline. And here we have a distinction, not really in terms of guidelines, but in terms of hospital practices. So we see that hospitals in Europe really prefer to use alkaline detergents. Whereas in the US, neutral pH enzymatic detergents tend to be preferred. So depending on which target market you have, we would suggest validating a different detergent and having a different detergent in your IFU. Again, what does that mean if you need actually both target markets? Well, we do know that for the US, they're more focused on the detergent being enzymatic than they are on it being neutral pH. And you do have detergents that are enzymatic and that are also slightly alkaline. So you could use a detergent like that to actually cover both of your markets with one detergent. But again, as we said for the cleaning method, this might depend also on what your hospitals are doing. And so you might want to look into this or connect with your user base to see what they are using, because it might be better to actually validate two detergents and have both an alkaline and a neutral pH enzymatic detergent in your IFU. But of course, again, this will take more time, it will cost more money. So that's really a business decision. Second step in our road trip is how to actually perform these cleaning validations. And here I want to talk about soil selection and acceptance criteria. So before I start, just to give you a very high-level overview of what we actually do in these cleaning validations. So we are going to soil the devices in a clinically relevant way. We're going to use a soil that mimics clinical relevancy, and we're going to soil the device in a way that mimics clinical use. Then we are going to clean the device as is instructed in the IFU in the instructions that you wrote. And then, as I mentioned, we do first a visual inspection, but then also quantitatively assess some markers such as the level of proteins, the level of hemoglobin. Now, in terms of soil selection, like I said, the most important thing is that your soil is clinically relevant. However, if blood is what is clinically relevant for you, there is again a difference between these markets. So for Europe, what we see is that regulatory authorities tend to prefer to use coagulated blood because they kind of see it as the worst case soil for blood. The US, on the other hand, they actually prefer defibrinated blood because they see that that one is worst case. So this can really be a tricky one if you want to validate both markets. So here it really depends on selecting a soil and justifying why you select that and why you think it's the most relevant soil for your device. And then in terms of acceptance criteria, this is where we have different guidance documents in different markets. So for the US, we have a standard called ST98 that lists different acceptance criteria for protein, hemoglobin, carbohydrate, or total organic carbon. In Europe, we also have a standard that we refer to, which is ISO 158835. Now, ISO 158835 is not necessarily a standard for validating reusable medical devices per se. It's really targeted towards validating washer disinfectors. But because it's really the only thing that we have, a lot of people refer to it for cleaning as well. Now, luckily, the action levels of both of these standards are the same. So for protein, for example, for both of them, the maximum amount that you can get is 6.4 micrograms per centimeter squared. The only exception here is Germany. So Germany has a separate standard for protein where they have more strict criteria. So they are looking towards 3 micrograms per centimeter squared. And for more critical devices, they even look at 100 micrograms per device. So here again, it's really just looking at what your target market is to decide which criteria you need and if you need these stricter criteria or not. So that brings us to disinfection. And again, I will first start with the IFU. And I want to talk about what happens for the intermediate disinfection step. And then I also want to talk about the disinfectants. Now, intermediate disinfection, what I mean by that is that you have both a cleaning step before and a sterilization step afterwards. So disinfection is really in the middle, but you do have that terminal sterilization step. So in terms of intermediate disinfection, for Europe, regulatory authorities will ask you if you have automated cleaning, and remember, if your device can withstand it, you have to. And if you have this process, you also have to include a thermal rinse. So you have to include a disinfection step as part of that washer disinfector program. This is really something that's more targeted towards the safety of the healthcare personnel, but this is definitely something that you will be asked for. For the US, this is not really strictly necessary. So you can just have cleaning and sterilization. There's no real need to include this intermediate disinfection step. It is something that is recommended in the guidelines as well, but they will not strictly enforce this. And that brings us to high-level chemical disinfectants. So here again, there's not really a difference in what the guidelines tell you to do, but there's really a difference between hospital practices between Europe and the US. So high-level chemical disinfectants, again, have different modes of action, sort of common modes of action. We have glutaraldehyde solutions, hydrogen peroxide-based solutions, or peracetic acid-based solutions. And here again, we have different products being preferred in different markets. So for Europe, we see that hospitals tend to prefer hydrogen peroxide-based or peracetic acid-based solutions, whereas for the US, glutaraldehyde solution is preferred. Again, what does that mean if you want to target both markets? Well, we are lucky here that there are some products available, such as Cydex OPA, which are widely available in both markets. So you could use those to have one product for both target markets. In terms of the actual validations and how to perform those, I want to go into a little bit more detail into thermal disinfection, which we also just talked about. So thermal disinfection, remember, you have to have it as part of your automated cleaning program for Europe. You don't really need it if you have a sterilization step in US. And that means that there's also a bit of a difference in how to perform the actual validations. So for Europe, you have to have it, but then because it's in your IFU, you also have to validate it, because everything that's in your IFU, you have to validate according to ISO 17664. Now for the US, if you do want to include thermal disinfection, but it's not your end process, so you still have sterilization afterwards, you don't have to actually do the validation. You only have to validate it if thermal disinfection is your end process. But the differences don't really stop there. So there is also a difference in how to then perform these validations if you need them. So for Europe, validations for thermal disinfection are always based on temperature. So we have temperature data that we use to calculate a lethality value. Whereas for the US, this is really going to be microbiological testing, where we use microorganisms and see if we can get an appropriate block reduction. So that already brings us almost at the end of our road, which is steam sterilization. Now, in terms of the IFU, there's quite a lot of decisions to be made when you're selecting sterilization parameters. And that has to do with the table that you see here. So as you can see, different hospitals in different regions, but also different countries in Europe, prefer different settings for their steam sterilization. So for the US, thankfully, the parameter sets are always going to be 132 degrees Celsius and 4 minutes of steam sterilization. Europe also mainly has 134 degrees Celsius. You do have certain countries such as the UK that go a little bit higher, but mainly it's 134. And then you see different times. So you have anything between 3 and 18 minutes of steam sterilization. So here again, it really depends on what your target markets are as to how to include or which parameters to include in your IFU. Now, you can design, let's say, worst case scenarios in your validation that will allow you to specify multiple parameters in your IFU. And I will get into that. But before I do, I first have to talk to you about how we actually do these steam sterilization validations. So how we design these validations. So steam sterilization validations consist of 3 tests. You have your sterility assurance level testing, and this is where we're going to use biological indicators, and we're going to make sure that they are killed off after testing. Second thing we're going to do is verify dry times, because we want to make sure that devices are dry when they come out of the sterilizer. Moist might result in reoccurrence of microbial growth, and we don't want that. Third thing we're going to do is temperature profiling. So we want to make sure that the temperature that is reached on the devices is the same as what we set in our sterilizer. So if we set the sterilizer to 132 degrees, we want to make sure that the devices reach this temperature as well. Now, for sterility assurance level testing, this is definitely something that you need for Europe and the US both. So you will always need to do this testing. Now, to come back to our parameter sets that I discussed previously, what you can do is select a worst case cycle to perform the sterility assurance level test. So we tend to approach this by using the lowest temperature and the lowest time, and then use that combined cycle to do the test. This is something that's really commonly done in the industry. There are some of our customers that have received feedback on this as to not do this and to run the cycle separate, but it's still considered to be current practice. So for example, if you want to validate all of US and all of Europe, what you would do is run a cycle at 132 degrees, so the lowest temperature, and 3 minutes, so the shortest cycle time. Dry time verification is something that is very much required for Europe and US as well. So equally for both of those markets, you have to do dry time. Now, this is unfortunately where we don't really have data that's going to show us which worst case cycles that we can use. Just because there's no correlation between cycle length and whether or not your devices are going to be dry easily or not. The way we tend to approach this is, for example, if you want to target both markets, run the US cycle separate, and then for Europe, take your shortest and your longest cycle and consider everything in between validated. So for example, for the 134 degrees parameter, validate the 3-minute cycle and the 18-minute cycle. And then temperature profiling is where we have a bit of a difference between markets. So for Europe, this is definitely required. It's definitely a must. You have to have the data. For the US, they sometimes will ask for it, but not always. And so we have seen customers where FDA has asked for temperature profiling, but they will not necessarily want you to do anything with it. They just want you to show that you have the data. For Europe, we tend to look at whether the temperature can reach -0 degrees and +3 degrees of the cycle settings. Now, so if you want to take a risk in your validation, you could leave out temperature profiling for the US cycle, but we would still do it for European cycles. And this is again where we would use the same approach as for dry time verification. So profile your shortest and your longest cycle, and then consider everything in between validated. So that brings us to the end of our road trip. I hope I was able to give you some information into how to design your road trip for your IFU and your validations, and give you some tips on where or which crossroads you have to make different decisions. But of course, if you still have any questions, don't hesitate to contact me. Here is my email address or our telephone number. So Nelson Labs is part of the Cetera Health Group with our partners Sterigenics and Nordion. And we also have our Cetera Health Academy, where you will be able to find this webinar, but also a lot more expert content. Thank you for joining.